Good afternoon, guys. Welcome. I want to give 5 minutes so that every other person will be here. Okay. Hello, everybody. How you guys doing? Sorry. I think I have to move back a bit. How many are we here? We are 52. Why? No. Okay. All right. That's fine. We have majority of the people here. So I'll just go straight to the point so that that's what we have for today. Okay. Yeah, no, it's fine. It's fine. It's fine. It's fine. You don't really need to comment. So, but the thing is, when I go over some slides, I'm going to ask questions, and I need everybody to attempt those answers. All right. That means you guys have to be very vigilant and pay attention. This is a very short discussion. We are going to have a very short discussion, but I want to throw some questions in. Since we are virtual today, we need to use the virtual advantage, to our favor. So what will happen is that when I discuss some slides, I'm going to throw some questions in here for you guys, and I need everybody to attempt it. I will need everybody to attempt. Okay. I think this works. And please, whenever it's lagging, you guys have to tell me, all right? Because sometimes I don't know. When I start talking, I just keep talking. Hello, welcome. All right. Let me jump into what we have for today. One thing we need to know is that nervous system is very important to everything we do, right? I am talking to you guys right now because my nervous system is working very well. Do you understand? You're able to talk, you're able to think, you're able to do things, you're able to feel things because of the nervous system. So the nervous system is a very complex system, right? One of the most complex systems because it controls virtually every other system. You understand? This is the foundation of our experience, personality, behavior, and this some people are going to specialize in neurobiology. Neurobiology combines that behavioral and life sciences together. All right. So this is more like an introduction. For this short part, we are going to describe the overall function of the nervous system and also describe its major anatomical distinctions. All right, the functional subdivisions of its anatomy. So I would like you guys to pay more than usual attention so that you'll be able to attempt these questions when I throw the questions. All right. Now, endocrine system and nervous system, they are relatives, right? But the major difference is that endocrine system, hello, welcome. The major difference is that the endocrine system, it communicates by means of chemical messengers, which are hormones, all right? Why nervous system does not use hormones? Instead, it employs chemical and electrical messengers, all right? For it to be able to send information from cell to cell. You understand? But hormones is purely what? Endocrine. So these are the types of nervous system because they can ask you, one of the following is not the things nervous system does. You'll be able to pick out the right thing it does from the options, all right? So sense organs receives information about changes in the body and external environments and transmits it. The CNS now processes those informations, all right? And when the CNS is done processing, it does what? It sends commands to the muscles. If I touch you, for example, it takes it to the brain, to the CNS. The CNS explains it, oh, that thing is a very romantic touch. Smile. Then it comes back and go to the specific muscle. You understand? Sometimes it's muscle of the face and you different muscles of the face and you start smiling. So the sensation I gave, somebody gave me by just touching my hand has triggered the other parts of my body, right? I start smiling, hair start going up. I know that, oh, this person really loves me, and this touch is touch of love. Now, how about when somebody hits you on the hand? What happens? You withdraw your hand. You get how? Because the sensory system takes the information to the CNS, and the CNS interprets it. Oh, that thing is very, very, very painful stimulus. You have to withdraw your hand. You have to frown your face so that the person doesn't do it anymore. I don't know if you see how it works. It's an interplay, right, between the entire muscles in the body and also the nervous system. All right. So virtually everything we experience is the nervous system. So there are two major subdivisions of the nervous system. We have the central nervous system and the peripheral nervous system. If I'm speaking too fast, please just put it in the box that I should slow down. All right. Just put it in the box. Tell me to slow down if I'm speaking too fast. But one of the advantage of this hour session is it will be uploaded on YouTube. So for people that need to recap, you can always go on YouTube, click the lecture and listen to it. You can slow it down if I'm going too fast. All right. So the CNS is made up of the brain and spinal cord. They will ask you this question, right? If you see a fish, I don't know, most of you eat fish in this here, all right? If you bring, there's one long bone in a fish, very long bone. That long bone you see in a fish is known as the vertebra, all right? If you touch your back, you see it at the back, the vertebra. Now, those vertebral bones, inside those vertebral bones, there are two things you see there. One, you see a thread-like substance. When you get home, just get a fish, long, big fish, open it, you will see the bone. If you take that bone, break it into two, what do you see? You see something like a rope. You understand? And fluid-like substances in it. I like it. I love it. Anytime I'm eating fish, I'm always eating that thing first before I even start touching the entire fish, because it makes sense. So that thing you see that looks like a cord, it's a cord. That's what we call the spinal cord. All right, it runs straight to the brain. Now, the fluid you see is something what we call CSF, cerebrospinal fluid. We are going to discuss it later on. All right. So it's very, very important that you guys know the enclosement of the spinal cord, and also the brain is enclosed by the cranium. All right. So the central nervous system is divided into two: brain and spinal cord. In case of if you're asked, now the peripheral nervous system is every other thing apart from the brain and the spinal cord. All right. What does it involve? Everything like the nerves, the ganglia. All right. They may ask you what is the nerve. A nerve is a bundle of fibers. All right, that is wrapped by a connective tissue. Then what is a ganglion? It's just the swelling in that nerve. All right. In that nerve, you see a big swelling. That swelling, that's where you see what? The cell bodies. So it's extremely very, very important that you understand this concept. Now, these are the two major subdivisions. So now we want to break them properly. All right. What you need to know when we are still in the overview, mind you, what you need to know is that the peripheral nervous system, for example, is divided into if you look at it, it contains the sensory and the motor, right? Sensory and the motor. That's the major divisions. Now, the sensory is the afferent. A before E. Afferent takes up, efferent brings down. That's what you should know. Afferent takes up, efferent takes down. That's fine. That's fine. It's okay. The most important thing is that you guys are here. All right. Now what am I saying? Okay, now the peripheral nervous system, the sensory part of it, right, which is the part that carries signals from the receptor to the CNS. It goes from the receptor to the CNS. It's divided into two. We have the somatic part and the visceral part. So the somatic part is the one that carries signal from your skin, bone, joints. I need you to understand this thing. Why you need to understand it is because they may be very specific about the question they want to ask you, right? Now, the peripheral nerve, remember we have the central and the peripheral nervous system. If you look at this slide, for people that we are not here, the central and the peripheral. The central is divided into brain and spinal cord. We are not discussing the central now. We are going to discuss that in Neuroanatomy 2. This is the Neuroanatomy 1. So we are focusing more on the peripheral. Why? Because the peripheral contains what? The nerve fiber, all right, which is the cell of the CNS. All right. Now that nerve fiber have a big part that has the nucleus, more like the nucleoplasm. We call it what? The ganglion. Now, we are focusing on the peripheral, as I said initially. Now, let's divide that peripheral. The peripheral has the one that takes up and the one that brings the information back. I hope it makes sense. So the one that takes up is known as afferent. That is the sensory. It senses and rushes to the brain. Afferent. Look at it here. It's the afferent division. All right. Now that afferent division. So let's keep the motor out first. Let's discuss the sensory, the one that goes up. All right. Now that sensory is divided into two, which is the somatic sensory and the visceral sensory. They may ask you a question. They may tell you one of the following, carries signal from the heart. That's the kind of question they may ask you. Then they will tell you somatic sensory, somatic motor, visceral sensory. I don't know if you get how they ask the question. I just got it. That's fine. That's fine. I don't know if you understand how they ask the question. So somatic, under the sensory, that's the afferent division, we have the somatic sensory. That somatic sensory carries information from the skin. You understand? From the skin, from the bones, from the joints to the CNS. All right? Now, the viscera, anytime you hear what? Viscera. Yeah, you're welcome. I see you. Anytime you hear the word viscera, you're talking about what? You're talking about organs. So the visceral sensory, you hear sensory, that's what carries the information up. I need you guys to get this into your head, all right? Now, the visceral sensory is visceral organ, sensory carries, means that they are what carries signal from organs. Somatic is body. That's muscle, bone, skin, joints. All right, that's somatic. Body. Somatic sensory carries information from this body to the CNS. Visceral sensory carries from the organs to the CNS. Organs like what? Like the heart, like the lungs, like the stomach, like the urinary bladder. Hello. All right. Now let's talk about the one that brings back. So if I want to send something to Brooklyn from Staten Island, I send it through what? I send it through the sensory, right? And if I want to bring it back from from Brooklyn back to me, it comes back through the motor. That's just what you need to know. Sensory, motor. Now, the second part is just talking about where is it carrying it from and where is it taking it to. Now, if you look at this particular page, the motor is the efferent. Efferent. It carries signals from Brooklyn, that's the CNS, to back to me, which I'm the effector. All right? CNS backwards. That is the efferent, which is also called what? Motor. Now, that motor is divided into two, which is the somatic motor and the visceral motor. Being somatic or being visceral talks about where it is taking that information to. Makes sense? I believe you guys understand that's what I'm trying to stress on this thing, because most of your exams is usually from the introductory part. Now, the somatic motor division carries signals to the skeletal muscles, all right? To the skeletal muscles. That's what leads to this contraction. See how my hand is moving, my mouth is moving. That's contraction and relaxation. All these things is the motor. You understand? The somatic motor coming to tell me, oh yeah, move your mouth, so that you'll be able to express yourself. All right. Now, the visceral motor is the one that brings the information back to the organ, carries to the glands, cardiac, and smooth muscle. Remember, cardiac muscle is in the heart, smooth muscle is in most of the organs of the body. So it just makes sense. All right. It just makes sense if you are able to, think these things. And another thing you need to know about this visceral motor division is it is autonomic. All right. Like you don't know it's happening, but it's happening. Signals is coming, the heart is pumping. Signal is coming, the heart is pumping. If there is a problem with the visceral motor division, the heart will no longer function effectively. The organs are no longer able to function effectively. That's how you need to think about this thing. Do you understand? That's how you need to think about it. No, not all somatic is automatic. No, not all of them. Now, that somatic, we have somatic motor. That's what I'm trying to tell you. The somatic motor is the one that carries information to the skeletal muscle. I move my hand because I want to move my hand. So it's involuntary. It's not automatic. But the somatic, the visceral division is the one that's automatic. Why? The visceral motor, innervates organs. Organ does not, your heart does not need you to tell it to beat for it to beat. Your stomach does not need you to tell it to contract for it to contract. All right. So the one that is automatic is the visceral motor. I need you to understand that. The motor division, the particular visceral. Okay, thank you. Yeah, you get it. Perfect. Okay, now that visceral motor, let's divide it. All right. We said it's automatic, right? Now let's divide it. There are two major divisions, which is the sympathetic and the parasympathetic. Yeah, listen to this. This thing is very straightforward, but it may be a little complicated to some people. That's why I want you to pay more than usual attention to this. Hold on. So that's why I need you to pay more than usual attention. How do I put it? Okay. Remember, we said the visceral motor. The fact that it is motor means it's coming back. All right? Coming back. So as being the Brooklyn is our central nervous system. It's coming back from Brooklyn to the effector, right? That effector can be skeletal or it can be organs. If it is skeletal, then it is what? Visceral. If it is skeletal, it is what? Somatic motor. Does it make sense? But if it is not skeletal, if it's not skeletal, it becomes what? Visceral motor. Now, that visceral motor can be in two places. It can be going to the cardiac or to the smooth muscles. Remember, there are three types of muscles. One is skeletal muscle, two is cardiac, and three is what? Smooth. The smooth is in all other organs. The cardiac is only in the heart. Then the skeletal is your normal skeletal muscle. All right? So that makes sense. Now, this visceral motor is divided into sympathetic and parasympathetic. Now, sympathetic and parasympathetic depends on what is it telling that organ to do. That's how this thing is divided. What is it telling that organ to do? Sympathetic is telling that organ, okay, activity, action movie, start moving, start doing things. That's sympathetic. Parasympathetic is, I need you to calm down. I need you to calm down. I need you to relax. All right? Relax yourself. Go and sleep. That's parasympathetic. Now, those effects also depends on which organs are we talking about. For example, sympathetic, if you are talking about the heart, you are talking about the breathing, the heart, the respiratory system, is activating it, it's accelerating it. That's why when you run, that's running, is usually sympathetic. When you run, what happens? Sympathetic comes into effect. All right, your heart is beating, beep, beep, beep. You are breathing fast, right? Because sympathetic is in effect. But as sympathetic is in effect, rest and digest, exactly. So as sympathetic is in effect, it is antagonizing some other organs. All right. So sympathetic activates the heart, and the respiratory system, but the digestive system and the urinary system is at rest at that moment. Why? It makes sense. Think about it. Fire, fire, fire. You start running. Your heart is pumping. Beam, beam, beam. You are breathing. You understand? But at that moment, you will not feel like peeing or pooing, like shitting, going to the toilet, or urinating. You won't feel those things. Why? Because the sympathetic will inhibit the digestive system and urinary system. Thank you very much. Somebody said we should use the word rest and digest. Do you understand? So the sympathetic system will activate, will make you be in activity. Like if you see all these people that play soccer or football, they see them running up and down, helter skelter. They are using their sympathetic nervous system at that moment. That's why their heart beats fast, they breathe fast. But you will never see any of them going out to go and urinate or going out to go and shit, because the digestive system is inhibited. The urinary system is inhibited. This is what they will ask you. So pay attention to this. And when you are talking about parasympathetic, at that moment, what is happening? You are resting. All right? When you are resting, your heart slows down, your breathing slows down. But what happens? Your digestive system starts working. Your urinary system starts working. That's why sometimes you see you wake up in the morning, you are running to go and piss, or urinate rather. And that's when you want to go to the toilet. You understand why? Because your digestive system works more when you are resting. So it's just an opposite effect. And I need you guys to remember this. That's why I'm spending so much time on this particular slide. I'm going to ask you some questions on it. All right. So if you look at it properly, look at this diagram properly. This is the spinal cord from the back. This is the brain. That's the CNS. You understand? And then this is the nerves and the ganglion. You understand? That's the PNS. Okay. This is a flow chart. I need you guys to use this flow chart as your wallpaper. I need you to use it as your wallpaper. If there's anybody on your wallpaper, remove that person. Tell that person, please, that you are doing an anatomy exam soon. All right. So use this as your wallpaper and try to memorize it to some certain extent. As you are memorizing it, try to understand. All right. Just a little recap on what we've discussed so far. This is CNS, brain and spinal cord. So this is out. This is PNS, peripheral nervous system, which involves the nerves and the ganglions, right? It's divided into two. One is the sensory, the one that takes information up. The second one is the motor, the one that brings information back. Now, the one that takes information up can take it from the skin, the bones, the joints, all right? That's what we call somatic. It can... Later we'll talk about that. Now, maybe 3 weeks of... 3 weeks. 2 weeks, probably. Now, the visceral is taking the information... from the organs, all right? Now, if you come to the motor is bringing backwards, all right? So it can bring it back to the skeletal muscle, S for skeletal, somatic. You understand? Just use the S for S. It's bringing it back to the somatic or skeletal muscle, all right? It's very, very voluntary. Now, this second part of the motor division is bringing it back to the visceral. That's the organs. The organs could be clothed by two different muscles, which are the smooth muscle and the cardiac muscle. All right. So it's affecting the heart and the other organs. All right. Now, this visceral motor, it depends on what is it telling the body to do or the other organs to do. Now, the sympathetic is telling everything. Oh yeah, activity, activity. But at the same time, the sympathetic is telling the urinary system to do what? To relax. It's telling the digestive system to relax. Now, this is the parasympathetic division. The parasympathetic division is telling your body, entire body, relax. The heart, relax. Respiration, relax. All right. But the parasympathetic is telling things like the urinary system, the digestive system. Oh yeah, activate. Go there. Do you understand? So I need you guys to bear that in mind. And we are done with that. Let me give you a question. And I will need everybody, please, to attempt that question. Everybody. Just a minute. Okay, it's a very straightforward question. This is more like testing the microphone. I think I'll give you very much question when we do revision. But testing the microphone, I need everybody. I need to make sure everybody is here. At least I should get up to more than 30 responses. Oh yeah, this is testing the microphone. Very fast. What is another name for the autonomic nervous system? Let me see the people that can get this. It's fine. If you are not sure, just write not sure, right? If you are not sure, you can write not sure. Even if you have anything in your head and everybody is typing their own, just put in your own answer. Everybody, please. Put in your own answer. You understand? Don't sit in the bed. Don't sit on the fence, please. I don't want sitting on the fence. Okay, you guys are the best. I think I've gotten up to 30 responses. You guys are doing well. So they say, what is another name for the autonomic nervous system? Autonomic nervous system is the visceral motor division or the visceral motor. Thank you very much. Visceral motor system. Yes. Visceral motor. Perfect. That's autonomic. It's what? It's what? Involuntary. You guys are very correct. Visceral motor. It brings back to the organs. All right. Bring back to the organs. Okay. So remember that. Now, this is another question. Which of the following, let me see, just hold on. Just want to give two questions for this beginning and we. Now, look at this question, guys. This is the second question I want to give, then we move to the next thing. Okay, this question, let me see. Okay, some dash neurons are specialized to detect stimuli, whereas dash neurons send signals to the effectors of the nervous system. The options: efferent, afferent. B. Okay, the option, the one in that box is A, efferent, afferent. The second one is afferent, efferent. The third one is motor sensory. The fourth one is efferent, inter. The fifth one is afferent, inter. Answer. Somebody say A and E. Come on, guys. Okay, all of you are very correct. It's the answer is B. A-E. A-E. Efferent. All of you are very correct. So let's proceed. You guys are so right. Thank you so much. I like the fact that all of you are here. We can actually do this thing another time. I think it's even more interesting because I'm able to give you some questions. Okay, so let's go. Thank you. Yes, AE. Afferent. If you see that in the exam, please don't fail it. Please. Okay, because sometimes it's a little confusing. Okay, so let's proceed. I hope I'm not lagging. Let's proceed. These are the universal process. Let's talk about neurons now. Remember, anything we are doing, we are breaking it down to the smallest level. All right, that's the tissues. Now, what are the universal properties of neurons? They will ask you. One is excitability, conductivity, secretion. All right, excitability, that's irritability, response to environmental changes and stimuli. Conductivity can respond to stimuli by producing electric signals and conduct them, send them to distant locations. Then this is secretion. All right. There are sometimes when electrical signal reaches the end of a nerve fiber, they do what the cell secretes a chemical known as what neurotransmitters. You need to know about neurotransmitters. Extremely important. Why is this not clear? Okay. Neurotransmitters are extremely important. Why they are extremely important for you to know? Yes, afferent, efferent. Why they are extremely important for you to know is because they help to transfer information usually from the nerve to the muscle. All right, like in the neuromuscular junction now, you talk about neurotransmitter like acetylcholine. And if there is something wrong with acetylcholine being able to match with the neuromuscular, with the receptors in the muscle, it leads to all these pathologies like myasthenia gravis, stuff like that. Like myasthenia gravis will even stop the ACE, that's the acetylcholine, from matching with the neurons there. All right, leading to flaccid paralysis. We have others that can cause spastic paralysis. Everything is circulating around that neuromuscular junction. But I'm not teaching you about muscles now. I'll do a video on muscles, and you guys will do well to watch it so that you understand it properly. Okay, so secretion is a part of property of neuron. Some people will be like, oh, what is this? Is it secreting neurotransmitters? All right. Okay. Now look at the functional classes of neurons. Pay attention. You will see questions on this. Functional classes of neurons. You understand? We have the sensory neurons, that's the afferent. We have the interneurons, and we have the motor or efferent neurons. The sensory detects, the motor sends back. Then the interneuron, what does the interneuron do? The interneuron, they receive signals from many neurons and do what? They integrate. They make decision. Oh, I touched you. The interneuron in the brain, where is the interneuron is specifically lying within the CNS, all right? And it is the majority of the neurons you have in your body. About 90% of the neurons you have in your body is interneurons. And what it does is that it connects the sensory and the motor. So it's the powerhouse of the brain. You understand? It sits down there. It's the headquarters. Do you get? You see, like New York now, I talk about Albany. That's where information is being passed through every boroughs of New York. All right? So that's the headquarters. So that's exactly what the interneuron is about. It sits down there, makes decision, tells you, you know what? That person that touched you, the person is interested in you. Do you understand? So it makes the decision. And once it makes that decision, it will now tell you, you know what? I want you to smile. It will send that signal out through what? The motor, that's the efferent. And the efferent will go and locate the specific muscle that has to react in accordance. Anything that is wrong with these things, the person will the person will not be able to have any sensation, feel any sensation. Right? That's how paralysis occurs. But we discussed that. Remember, this is anatomy. Anatomy is what? Location, location, location. And also function, function. Physiology is function, 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 rather. Anatomy is location, 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 or structure, structure, structure. So I need all of you to be aware of this. All right? Okay. People that have not subscribed, please do well to subscribe. And put a like, click this hand. That one that they did, like, click it. Okay, let's continue. I don't want to get distracted. This is the diagram. All right. This is sensory. You see, you touch something, you feel the thing. The sensory takes it to the CNS. In the CNS, you have interneuron. Then interneurons says, okay, this is a very good, go back to the muscle. Efferent. It's very direct, very straightforward. All right, I gave you a question on it. Now, let's talk about the neuron. Remember, the neuron, as I told you, is the neuron, as I told you, is a cell. All right, so it has everything that a cell has. If you think about a cell, just like when we talk about muscle fibers is a cell of the muscle. The same thing, neuron is a cell of the brain. All right, so think about it in that direction. Ask yourself, what does a cell have? A cell has what? The control environment, which is the neurosoma. You understand? So the neurosoma is the control center of the neuron. All right, also called what? The cell body. Neurosoma is also called what? Cell body. Single and centrally located. So a neuron, a nerve cell has only one cell. It only has one nucleus, rather. That's what I wanted to mention, one nucleus. But if you think about the muscle fiber, that's a muscle cell. It's muscle cell. If you bring one muscle cell, it has a lot of nucleus, unlike this nervous cell. So these are little differences that you can remember. You can try to remember. So a neurosoma has a single centrally located nucleus with a very large nucleolus. That's the surrounding environment. Now, it has a cytoplasm. Remember, it's a cell. If you enter a cell, what do you see? You see the cytoplasm. That's the internal environment of that cell. All right? So that's exactly the way the neuron is. If you enter inside, you see the internal environment, that is cytoplasm. Then you see the nucleoplasm. That's where you talk about the nucleus. But now, outside the nucleus, let's talk about the entire cytoplasm. What do we have there? You have the mitochondria, the lysosome, the gum complex, and also inclusion bodies like fats, lipid droplets, melanin, lipofuscin pigment, and all those things. All right, those are inclusion bodies. Every other thing is an organelle. Rough other organelle, endoplasmic reticulum, cytoskeleton. All right, but bear in mind there is no mitosis, no centrioles. So these two informations are very important because they usually ask it in your exam. All right. Now, the dendrites is just the branches, just like the hair. Assuming I have long hair, it would have been the dendrites for me. All right. My face will be the neurosoma, where the nucleus is. All right. Then this part will be the axon. So I need you to put it in your head that way. So we have what? What do we have? We have, the cell body. That's the neurosoma. This is the neurosoma. That's one. You have the dendrites. You have the axons. Neurosoma, the dendrites, the axons. This is what makes up a neuron. We've talked about the neurosoma. We've talked about the dendrites. The dendrites is just the branches that come off the neurosoma. So if my face is the neurosoma, then my hair is what? The dendrites. That's the branches. And it's very, very important because it is the primary side that receives signals from neurons. Neuron to neuron transmission, it transmits it to the dendrites. It receives those information. So the more dendrites a particular nervous system has, all right, a particular neuron has, the more dendrites a particular neuron has, the more information that neuron can receive. It's an inverse proportionality. All right. Now, you talk about the axon, which is down. So as you just use yourself to visualize a neuron, and you'll be able to know what is dendrites, what is neurosoma, and what is axon. All right. Now, the axon is the nerve fiber itself, downwards. All right. It originates from the mound of the neurosoma called the axon helix. So this is the neurosoma. This is the dendrites. I have a very long hair. Can you see the long hair? Yes, all of you can see my hair. So I have very, very long hair. So this, my very long hair, is the dendrites. This is the cell body. This is the mold, axon helix, and this is the axon downwards. All right. So the axon, these are everything you can see in the axons. Specialized for rapid conduction of signals to distant points. Exoplasm, that's the cytoplasm of the axon. Exolemma, the membrane of the axon, and also something we call the myelin enclosure. That's the myelin sheet. All right. It helps for faster transfer of information. I'm going to discuss it later. All right. Now, the end part, we call it terminal arborization. All right. It's just a complex fine fibers or fine branches. The terminal arborization. Then we have the axon terminal itself. That terminal is where it synapses. It can either synapse axon to one neuron to another can synapse. All right. One neuron another can synapse, or that neuron can synapse with a muscle, which we call neuromuscular junction. So we have neuronal junction and we have neuromuscular junction. neuronal, when two neurons are, are synapsing. We discussed that. Then neuromuscular, when two neurons are, when a neuron and a muscle is what? Synapsing. There are things there are intricacies that are involved, which we will discuss. Remember, we are still in the introductory phase of this discussion. Now, if you look at this, this is a beautiful structure of a neuron. All right. You see the hair. You see that my head I was telling you about? That's the dendrites. So if they give you this kind of diagram in exam, you eat it. You eat this diagram. You understand? And show them what you are capable of doing. So you label this part what? Dendrites. Sorry. I told you, think of my face as what? This is my very big face. All right. As the neurosoma. All right. So this is the neurosoma. Look at it. The head, the neurosoma. The hair is the dendrites. Then the neurosoma, what do you see? You see the soma, you see the nucleus, you see the nucleolus. All right. Now, coming downwards, you see my neck. What did I call it? I called it what? Axon hillock. See it here? The mound. All right. Now, from that axon hillock down is the axon. I played too much. So this is the axon down like this. All right. So if you look at it very well, you see this is the one node, this is another node. All right. There are the internodes. Right. If you come down, what do you see? This is the axon terminals. You see the axon terminals? Then this last part of the axon is known as what? Terminal arborization. You see this is the terminal arborization. So for this now, this place will be terminal arborization. Why this place will be what? Axon terminals. Now, this is myelin sheets. All right. This is myelin sheets. Created by the Schwann cells. We are going to discuss it. Now, this node and another node, all right? In between them is called what? Node of Ranvier. This is an internode, this is an internode rather. Then in between them is called what? Node of what? Ranvier. This is node of Ranvier. So they can give you this diagram in the exam and tell you to call this. They call this A, call this B, call this C, call this D. You should be able to understand it properly. All right? Okay. Now let's talk about the structure of the neuron properly. Oh, no, that's fine. That's fine. As far as you can listen to me, I'm good to go. Okay, so we have the structural classification of neurons. Structural classification of neurons. This is a very nice diagram that you should also know. Now, we have a multipolar neuron, we have bipolar neuron, we have unipolar neuron, and we have anasonic neuron. Please, you have to pay attention so that you can catch this thing. Afferent, efferent. Yes, you're correct. Now, multipolar neuron is the most common. If they ask you which is the most common, please tell them that it's the multipolar what? Neuron. It's the most common in the CNS. All right? Multipolar. One axon, multiple dendrites. Multipolar. One axon. This is one axon, right? Then this is dendrites. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this is a lot of dendrites. This is one axon. So this is a multipolar. I hope we are following. Now we have the bipolar. Bipolar is one axon, one dendrite. One axon, one dendrite. This is one bipolar here. Look at it, guys. One axon, one dendrite. One axon, one dendrite. One axon, one dendrite. You see, the dendrite did not originate from this is multipolar. Look at multipolar here. This is multipolar. 1, 2, 3. All right. This is the axon. This is one dendrite. This is another offshoot of dendrites. That's multipolar. Now, this is bipolar. 1, 1. Now, listen, they may not give you this diagram, but there is the special information you need to know. The special information you need to know is that they will ask you which type of neuron is found in the retina. See this retina? Which type is found in the retina? Which site is found in the olfactory cells? Which site is found, in the inner ear? That's what they're going to ask you. And you tell them it's bipolar. And when they ask you what is bipolar, bipolar is one axon, one dendrite. Multipolar is one axon, multiple dendrites. Now you have unipolar. Unipolar, single process leading away from neurosoma. Sensory cells from the skin and organs to the spinal cord usually have unipolar. They hardly ask this, but they ask this and this most times. Then we have the anazonic. Anazonic. Many dendrites, no axon. Anazonic. It does not have axon, but it has a lot of dendrites. You can also see it in the retina, you can see it in the brain, and you can see it in the adrenal gland. Apart from these places, every other part is what? Multipolar. Straightforward. I'll give you a question and you understand what I'm talking about. Later, you look at these diagrams. Let's not spend too much time on it. Okay, I think I should just give you a question right now. Let me see how many people are paying attention. Let me see people that are paying attention. Let me give you a question. Look at this question, guys. Okay, are you ready? I believe you are ready. Look at this question. Adrenaline is rushing, right? Okay, take it. Let me see. Let me see who was listening and who wasn't. About 90% of the neurons in the nervous system are dash neurons. Somebody said I should never catch them slacking. No, you guys are too hot. You understand? Like, you guys are too active, and I love it. I love it. Okay, keep going. Let's see. Okay, somebody said more. Okay. Okay, not sure. You guys are very, very good. The answer is interneurons. The answer is what? Interneurons. Like, if you look at this part, you see, this is the beauty of this team. I'm beginning to like it, though, honestly, because I am able to navigate. You see, look at the question. Look at the question, guys. Interneurons, about 90% of all neurons. You understand? You remember, usually the most standard exams are multiple choice questions, right? So it's in between the line. You have to read in between the line. So let me ask you this other question. Let me ask you this other question. Truth of the matter is that you are not master's anatomy students, so your question will be very superficial. What is the most common type of neurons? I'm expecting everybody to give me this answer. Like this, like this, like this. Keep going. Everybody. This one is everybody. Nobody's not sure. I am not expecting any not sure here. Perfect. That's great. That's great. Then let me ask you this particular question. Okay, no, the answer is multipolar, guys. The answer is multipolar. Now I need you to answer this question, guys. Most metabolic and regulatory functions in a neuron happen where? Okay, I see you. You say fat fingers. You're funny. So most metabolic and regulatory functions in the neuron happen where? The options, I should give you options. Okay, let me give you options. The axon, it happens in the axon, the cell body B, the dendrite C, the axon hillock D, the Schwann cell E. The axon, the cell body, the dendrites, axon hillock, Schwann cell. Keep going, guys. Cell body. Please, the answer is cell body. Please, there is this question that I just thought of now, and I really, if I don't answer this question, I can choke. And I know the answer is cell body, guys. Yes, the cell body. No, not axon. Cell body. Cell body. Virtually everything happens there. It's just like the nucleus of the entire thing. But I want to ask you this question. If I don't ask the question, as I said, I may choke. Let me ask you this question. Okay, let's go. The term nerve fiber refers to A or and dash. Let me give you options. I think I should give option. A, nerve fiber. It refers to what? A, nerve cell. B, organelle in the nerve cells. C, organ. D, axon. E, bundle of macromolecules in a nerve cell. A, nerve cell. B, organelle in the nerve cells. C, organ. D, axon. E, bundle of macromolecules in nerve cells. Keep going, guys. Keep going. Okay, guys. So if you answer axon, you are correct. If you don't answer axon, please, the answer is axon. Look at this page again. You see the dendrites, you see the axon, you see the brackets. Axon is nerve fiber. Axon, nerve fiber. Axon, nerve fiber. Then we have the dendrites, we have the neurosoma. These are the contents of the axons. All right. Okay. Okay, so that's good. So let's move. Let's move. I don't want to waste too much time here. Let's fly before the time chokes. Now, guys, I want you to talk about the supporting. Let's talk about the supporting cells. Supporting cells, that's the accessory cells. Remember, whenever we are talking about everything, we are talking about accessory. Make sense? Digestive system, we talk about accessory. Urinary, we talk about what? Accessory urinary organs. So here, let's talk about the supporting, that's the accessory cells, all right, in the nervous system. We call them, these accessory cells, we call them supportive cells or neuroglia. So anytime you hear neuroglia, you are talking about what? The supportive cells. I know my accent is terrible. That's fine. I know you guys can hear me at the end of the day. So let's talk about these supportive cells. The supportive cells, how many of them? There are about 1 trillion neurons in the nervous system. The neuroglia outnumber neurons by at least 10 to 1. So if there are 1 trillion neurons, that means the neuroglia is like what? 10 trillion neuroglia. Do you understand? Or even 11 trillion, because it outnumbered it by 10 to 1. Okay, 10 million. 10 trillion rather. So, but my point I'm trying to make is, I want you to see the neuroglia as support to the neuron. The neuron is the cell itself, but the neuroglia is the support of that cell, all right, in the nervous system. And it's a lot, meaning that if there's a problem with the neuroglia, there'll be a problem in the nervous system because it's a lot, all right? Now, the neuroglia is the same thing as what? Glial cells. So it's a different type of cell from the neuronal cells. Neuroglia, glial cells, neuroglia, glial cells found where in the neuron. What do they do? You must know these things, guys. One, they protect neurons and help them function. You understand? They bind neurons together and form framework for nervous tissue. In fetus, they guide migrating neurons to their destination. They serve as chaperone. Now, if mature neuron is not a synaptic contact with another neuron, it is covered by what? Glial cells. So if this is a neuron and this is another neuron, and they are not connected to each other, what happens? This place is covered by the glial cells, this place is covered by glial cells to protect them, all right? So it serves as a very strong protective function, all right? And moreover, it helps to bind that neuron. It binds it, wears it as a cloth, protects it from anything that can happen to it. And in fetus, we know what it does, all right? Okay. Now, what are the types of neuroglia cells? What are the types? Because you need to understand them, especially if you look at this diagram, but let's discuss before we jump. 4 types of glial cells: oligodendrocytes, ependymal cells, microglia, astrocytes. Close your eyes and put it in your head. Quickly. Close your eyes. Oh, yeah. Say it out. Talk. Don't worry. It's okay. I can hear you. Talk. I can hear you through the spirit. Talk. What are the 4 types? Just say it out. You don't need to type it. Say it out. I'll hear you. Okay, I heard you. Some people did not talk. I know you didn't talk. You did not say anything. I did not hear your voice. Okay, guys, let's he said not sure. Okay, that's fine. That's fine. Some people, I think their screen is lagging. I'm sorry. Maybe if you are not catching up after this stuff, try to listen to the lecture. Now, there are 4 types of glial cells, all right? We have the oligodendrocytes, we have the ependymal cells, we have the microglia, and we have what? Astrocytes. Oligodendrocytes, ependymal cells, microglia, astrocytes. But it's not enough for you to just know. It's not enough for you to just know the names. All right? It's not, no, I'm not asking the question. I said, what are the 4 types? I said you should learn the 4 types of glial cells, all right? And close your eyes and say it out. And I didn't hear you, but I heard every other person. Okay. Oligodendrocytes, what do they do? Guys, the equation will majorly be on their function. What do oligodendrocytes do? They form myelin sheets. In where? The CNS. You see this thing, let me make it dark. They form the myelin sheets in CNS. They will ask you, what forms the myelin sheet in the CNS? What forms the myelin sheet in the PNS? You get it? So oligodendrocytes form myelin sheets in the CNS. And what is the function of myelin sheet? To speed up signal conduction. It's just like in the digestive system, you see enzymes. What is the function of enzymes? Enzymes is just to speed up chemical reaction. In the brain, the oligodendrocytes, specifically in the brain and the spinal cord, that's the central nervous system. Oligodendrocytes will sheet the myelin sheet, the nerve cell, and we help it to conduct information what faster. So it serves as an enzyme. I'm just saying that, don't mind me. Okay. So what they usually do is they wrap around nerve fibers, all right? If you look at these guys, look at this diagram. You see the wrapping. This is the myelin sheet. This myelin sheet. If this thing is in the CNS, that means they will ask you if this thing is found in the CNS. This myelin sheet, where is it produced from? Oligodendrocytes. All right. Now, the second one is ependymal cells. Ependymal cells. Ependymal. It's just sounding like epithelium, right? For people that did tissue properly, epi. So what does it do? It lines cavities. It lines cavities like sheets covering cavities. It's the epithelium of the brain. Simple. Chicken, huh? Do you understand? It's the epithelium of the brain. So ependymal cells are the epithelium of the cell, the line cavities of the brain. And since the line cavities of the brain, they help in secretion of what? CSF. They will ask you, what secretes CSF? Which type of neuroglia secretes CSF? It is what? Ependymal cells. Which type of neuroglia in the CNS produces myelin? It is what? Myelin sheets. It is what? Oligodendrocytes. Chicken, you are good to go. Now, the microglia, what are they doing? They are majorly there to repair, to solve the problem, all right? The problem can be the deposition of debris. The problem can be damage in the nerve cells. The microglia develops what? White blood cells, that's monocytes. And when it develops white blood cells, it will clear up that debris, helps for, repair of damage. It comes to that place where there's a problem and concentrates itself. It's just like when you have an injury now, a cut on your hand. What happened? The blood starts flowing more there. Why is the blood flowing there? Because the blood is bringing both platelets, bringing white blood cell, leukocytes, so that it will mop up the infection that is there for it to heal properly. That's exactly what microglia does. Chicken, you don't need to stress yourself. Now, when we talk about the astrocytes are the most abundant glial cell in the CNS. Listen, anytime you hear most abundant, that's the kind of question they like to ask. They may ask you, oh, which type of neuroglia is the most abundant in the CNS? You talk about astrocytes. You remember when we talk about most abundant again? Do you remember? We mentioned interneurons, 90% of it. Then we also talk about what? Multipolar. All these things are markers. These things are things you should, be able to understand and memorize. So the most abundant in the glial cell is what? What happened? the number dropped from 70 something to 40 something. Yes, everybody. Okay, let me just continue anyway. So the most abundant glial cell in the CNS is the astrocytes. And what does it do? It covers the surface, right, of non-synaptic regions of the neurons in the gray matter. Now, it has a lot of function, diverse functions. It forms the supportive framework. It has extensions that helps for blood-brain barrier so that things does not enter the brain. All right. I'm still having 43 people here. We shouldn't be 43. We should be up to 70 or 100 here. They monitor neuron activity and regulate blood flow to match metabolic functions. They convert glucose to lactate and supply this to the neurons. They secrete nerve growth factors, and they do a lot of things, all right? That's why if they have a problem, you call them what? Astrocytosis, or you can even have a sclerocytosis. That's when neurons are damaged. Astrocytes form hardened scar tissues and fill in the space. That's astrocytosis. So it's very, very important for you guys to know everything about astrocytes, all right? So if you look at this diagram very well, what do you see? You see a neuron. This is a neuron, all right? This is a neuroglia of the CNS. What are you seeing? You see, this is an astrocyte. That's really important. Remember what astrocyte is. It has a diverse function. It does a lot of things, all right? And what do you see? This is microglia, right? Microglia serves as the defense system to mop up any debris and also to help in repair. Now, this is oligodendrocyte. You see, this is oligodendrocyte. This is how it's now giving myelination to this nerve cell. See, giving it myelin sheet like a rubber covering this place. So what happens when information is coming down now? The information does not need to pass through this rubber part. The information will just jump, gallop, jump. Before you know it, it has gotten to its destination. So the idea of this myelination is for it to be what? Faster. All right? For it to be faster. You have to tell everybody to come be active here, because if they are not, that means we may not be doing this thing next time. Okay. Now, there are other types of glial cells, but these glial cells are specifically found in the PNS, peripheral nervous system. Example, the Schwann cells and what? The satellite cells. The Schwann cells and what? The satellite cells. So we have the... No, you are not. You are just for something here. I'm expecting up to 80 people here. So there are two types, only two types of glial cells in this PNS. Remember, all these things are in the CNS. We have the oligodendrocyte, the ependymal, the microglia. Remind me of octopus. Yes. yes, that's the oligodendrocyte. I believe that's what I'm talking about. So we have the oligodendrocyte, ependymal cells, microglia, and astrocytes. I believe all of you already know what this thing does. Just put it in your head. You don't need to go and review after this class. This is your review. Except when we are doing the major exam review. Now, for this PNS, that's peripheral nervous system, we have the Schwann cell and we have the satellite cells. The Schwann cells, what do? These Schwann cells, they envelop the nerve fibers in the PNS. They produce what? Myelin sheets. So this, if you see oligodendrocytes, know that they are talking about what? CNS. If they tell you, so you need to pay attention to your question. They may be asking you, which one produces myelination in the CNS? Oligodendrocytes. But if they ask you, which one produces myelination in the PNS? You are talking about what? Schwann cells. So you have to be focused. I don't want a situation where you see a question. They say, oh, which one causes myelination? You will not even look at where. You won't look at where. Piam! You have answered. It's just like in the US. They build houses here with I don't know. I think this thing sounds like wood. But in Africa, we don't build houses with wood. You understand? Because that place is hot. Instead, we build with bricks. All right? So that's how it is. One is CNS, one is PNS. If Africa is CNS, for example, that means you build with bricks. That's oligodendrocytes. If America is PNS, that means this is what? Schwann cells, wood. Do you understand? Like, just look for a way to understand these things, and you won't need to remember at all. Also, it assists in regeneration. Did you see? This assists in regeneration of damaged cells. So it doesn't just, for Schwann cells does not just produce the myelin sheets, it also assists in regeneration. Why? Because for the CNS, the CNS has the astrocytes, the CNS has what? The microglia that helps in all this damage control. Then that's why the oligodendrocyte is very specialized to conduction. But in the PNS, It is not specialized to just Schwann cell is not specialized to just conduction. Instead, Schwann cells helps in repair of damaged tissues. Now, finally, for the glias, we have what? The satellite cells. The satellite cells, they surround the neurosoma in the ganglia of the PNS. They surround the neurosoma. If they surround the neurosoma, what do? They provide electrical insulation around what? The neurosoma. They regulate the chemical environment of the neurons. You need to remember the satellite cells. Satellites. You know, they insulate everything in the neurosoma. They insulate it. Why? Because that place is the center. It is the center, headquarters. I believe all of you understand this thing. All right. I believe everybody understands it. So what you do for me, you do a short exercise for me very fast. Everybody here, before I proceed, everybody needs to do it. I want you guys, don't look at the slide. Don't look at the slide. I want you to just in one line, in one sentence, don't put it one after the other. I want you to give me the 4 for CNS. Just write CNS, put the 4 of them. Then PNS, put the 2 of them. If you don't remember, that's fine. If you remember only 1, put it there. If you remember only 3, put it there. But I want you to type it now without looking at your books. Everybody, let's go. Just type it in the comment section without looking at your books. Just anything you can remember. Anyone you can remember. There are 6 of them. 6 of them. So if you write CNS, you type, then continue from that line, PNS, you type, or whatever works for you. But please don't look at your book. I want you to see what you have in your head right now. Don't look at your book, don't. Don't look at your slide, guys, don't even look at what you wrote down. Don't look at what you wrote down. Just put in something that you can remember. Don't look at what you wrote down. You have to be very honest to yourself, because I don't know. I'm not there. But let me look. Let me use my spiritual eyes. Let me turn in my spiritual. Somebody is looking at his book. Don't look at your book. Let's go. Even if it's 1, you remember, that's fine. But I need you to test yourself. Okay, you guys are the best. You guys are doing quite well. Let me see. Somebody said Schwann satellite. Okay. Let me see. Oligodendrocytes, astrocytes, PNS, CNS, astrocytes. Okay, you guys are great. So what you need to do now is, I'm giving you 30 seconds. Go back to it and look at it again, and make sure it enters your head. What do they do? All right. Make sure it enters your head. Make sure it enters your head. Quickly. I think I can throw in one question. I know you guys want to answer a lot of questions, but I'm not teaching a lot of questions today. But let me throw in 1 or 2 questions for you and see. Perfect. Okay. So I want everybody to attempt this question. Which glial cells, yes, attempt this question very fast. Which glial cell is found dropped around axons in PNS? Let's go. Okay. You guys are very active. I love it. More than 50 people are being active here. Okay. I think we'll be doing this thing many times. Okay. So you are correct. The answer is what Schwann cells. Let me see if I can chip in one more question. Let me chip in one more question. Look at this question. What is the primary? Yeah, you guys are correct. The answer is Schwann cells. You are absolutely correct. Look at this question. What is the primary site on a neuron for receiving signals from neurons, from other neurons? What's the primary sites? Let's go there. Let's go there. What's the primary site? What's the primary site on a neuron for receiving signals from other neurons? You guys are absolutely killing it. It's the dendrites, all right? Dendrites is the answer. So that means you guys are following. Okay, look at this question. Which cells from myelin sheaths, okay, which cells form myelin sheaths in the spinal cord? Which cells form myelin sheaths in the spinal cord? Perfect. Keep going, guys. No. I'm not saying perfect. I won't tell you if you're right or wrong so that people will express themselves. Keep going. You guys are correct. Extremely correct. The answer is what? Oligodendrocytes. You see, I like the fact that you guys are getting this thing very fast. Oligodendrocytes. Okay, so let me proceed. Now let's talk about myelin sheath very fast. Let's talk about myelin sheath. Myelin sheath is insulation, right? Formed by what? In this oligodendrocyte in CNS and Schwann cells in the PNS. We already discussed this. What does myelin sheath contain? It contains 20% protein and 80% lipid. 20% protein, 80% lipid. Put it in your head. You see this percentage? You need to know it. Please, for the love of God. Now, myelination is production of myelin sheath. We know that. This is everything about myelination when it begins from 14 weeks of fetal development. That's in utero when the baby is still 14 weeks in the mommy's tummy. You know, it's produced rapidly during infancy, completed in late adolescence, and dietary fat is important for CNS development. Right? So when a child is growing, still growing up, you know, you can be giving that child a lot of dietary fat to make sure the child has a proper development of the CNS. All right. So, yeah, that's it. That's for myelination. All right. Let me see. So this is just discussing about the things and the about the Schwann cells. It lays down as many as 100 layers of membrane. It does not have cytoplasm. You know, there's something we call neurolemma, which is the thick outermost coil of the myelin sheath. They may ask you about neurolemma. Let me tick it. Let me make it thick. They may ask you about neurolemma, which is the thick outermost. If you just see thick outermost part of the myelin sheath, you are talking about the neurolemma. Okay. So look at the myelin, the myelin sheath in the PNS. Mind you, this is the PNS. Because it's the PNS, what do we have here? This is the Schwann cell. You see it? This is the sheath itself. This is the Schwann cell. All right? This is the axoplasm and everything. This is the neurolemma, the outermost part. This is the neurolemma. See it? Neurolemma. The part that is covering this nucleus. See it? Okay. So nothing much. So in the CNS, oligodendrocytes, we already discussed that. Oligodendrocytes anchored to multiple nerve fibers cannot migrate around one of them like Schwann cells. So the difference between the CNS, that's the oligodendrocyte, and the Schwann, is that the Schwann cell just wrap around just one. They myelinate multiple axons at once, unlike perfect, you're correct. Thank you so much, Catalina. Thank you so much. So you see the difference? This Schwann cell only wrap around one, but this guy here is anchored around what? Multiple nerve fibers. All right, cannot migrate around any one, unlike just like the Schwann cell. All right, must push newer layers of myelin under the older ones. So myelination spirals are inward toward nerve fibers. Now look at the diagram. You see it? Just one oligodendrocyte is producing this myelin sheet, this myelin sheet, this myelin sheet, just like that. So it's multiple nerve fibers, not just one. And it's producing new one and put them under the old ones. You see how it circulates. That's how it circles it. Okay. So this is segmentation, all right? When you're talking about segmentation, nodes of Ranvier is the gap. We already discussed that. Internodes is myelin covered segments from one gap to another. That's internode. In between them, you see the nodes of Ranvier. Then we have initial segmentation. We have the trigger zone. The trigger zone, the axon hillock, that's that neck I was telling you about, and the initial what? Segment, right? They play a very important role. Why? Because that is when nerve signals are being initiated. Nerve signals are action potential. This is what you need to know. All right? Very, very important. Now, we talk about tumors. Remember, this is not pathology. This is pharmacology. This is physiology. What am I saying? What am I saying? This is physiology. All right? Physiology is normal. Normal. I am talking now. That is a normal thing. All right? Anything that makes me not to talk, you see me one day on the road and you say, hey, professor, how are you doing? I'm not able to talk. That is what pathology becomes abnormal. So this is abnormal as well. All right? Now, talking about the pathology, talk about tumors. What are tumors? They are just rapid dividing cells. All right? They grow so much. But bear in mind that mature neurons, they have little or no capacity for mitosis, and they seldom form tumors. Funny enough, I want to tell you that most dangerous tumors in the brain are tumors that came to the brain, not the tumors that developed in the brain. That's one information you need to put in your head. They are the tumors that migrate. Metastasis. Tumors can metastasize. Lung cancer, for example, can metastasize to the brain, bone, and all those things. So if they come to the brain, that is a problem. But you see those ones that grow in the brain, they are hardly a problem. That's just the thing. So brain tumors arises from meninges, protective membranes of the CNS. Metastasis from non-neuronal tumors in other organs. You see what I'm talking about? Either from the meninges, that's the protective areas, or from non-neuronal organs. All right? Another thing that produces tumors in the brain is what? The glial cells. Remember the glial cells? There are a lot. There are 10 times the number of neurons we have. All right? So even if those neurons are not growing tumors because they are not metastatic, they are not mitotic rather, all right? The glial cells are mitotic all through their life. They are actively growing, actively dividing, all right? And that's why we have a lot of what? Gliomas. So if you see glioma, they have grown rapidly and they're highly what? Malignant. Gliomas, all right? So blood-brain barrier is also very important. Remember, blood-brain barrier that is formed by what? What forms the BBB? Guys, what forms the blood-brain barrier? Or what produces them? Can you remember? Let me see if I can get up to 5 correct answers. Put it in there. They say, what produces this blood-brain barrier? Let me take you guys back, because everybody seems to have forgotten. Ependymal. Look at it. It produces the barrier that secretes that fluid. Look at it. Ependymal cells, they line cavities. Look at it. They line cavities. Extremely important that you know this. All those cavities of the brain, they line it. So it's like they shield the brain from anything that is coming in. All right? They shield the brain, they line it. And I forgot to mention something. Thank you that I have to go back to this thing. Look at this thing. If they ask you what is the epithelium or the ependymal cell of the brain, it is cuboidal. Remember, guys. I don't know if you are part of any class. I've been able to, if you look at tissues, I watch a tissues video. Maybe after this, go and look at tissues video. The tissues video will tell you more about the epithelium. All right. Now, I mentioned something that any epithelium that produces fluid is usually cuboidal. That's why major epithelium of the urinary system is cuboidal. Now, when I was making that video, somebody asked me a question. Somebody was like, so why is it that the ureter is transitional? I told the person, the ureter and the bladder. I said, the ureter and the bladder, they don't produce fluid. Instead, they accommodate fluid. They carry fluid. So that's why they are not cuboidal. That's why they are transitional. All right? Like the kidney also have other epitheliums, like the simple squamous. You feel the simple squamous in the descending, that thin descending loop is simple squamous. You understand? There are two major places you see simple squamous in the body. That thin descending loop of the kidney and also the alveoli of the lungs. Those are the two major places you see simple squamous. Simple squamous. Those are the two major places you see them. The two major places you see them. And usually you see simple squamous because it allows transfer of substances in between it. All right. So that descending loop allows water to reabsorb into the bloodstream. Water reabsorption or water conservation. So anyway, let's not distract ourselves from what we are discussing. Let me continue. Where am I? So a lot of gliomas. So BBB decreases effectiveness of chemotherapy. Trust me. All right. That's why people transition to radiation and to surgery. Okay, let's go. Now we have degenerative diseases. This degenerative disease is the disease that affects what? Myelination. Do you remember what the function of myelination is? Myelination happens so that it can cause faster transfer of information. So if the myelin sheet breaks down, what will happen? The information will not process fast. Haven't you seen individuals where you ask them questions and they don't know what you are saying until some certain time passes by? They are slow. Yes, it's less electric signals, so they are slow to respond. They don't see properly. All right, they are having double visions. They don't talk well. Their speech is, the speech effectiveness is reduced. All those things is as a result of what? A breakdown of myelin sheet. So if you look at this now, the major problem is what we call what? Multiple sclerosis. That's the number one problem usually we have with myelin sheet degeneration. Multiple sclerosis. Now, the multiple sclerosis, oligodendrocytes and the myelin sheets in the CNS, they deteriorate. This deterioration causes multiple sclerosis. Multiple breakdown. That's what they're telling you. Why? Because oligodendrocytes, it insulates different neurons. You see? It insulates different neurons. If you go back to this diagram, you see it. This oligodendrocyte insulate this neuron, insulate this neuron, insulate this neuron. So if anything, if this guy start breaking down, it will affect this guy, affect this guy, affect this guy. That's why we call it what? Multiple. Oh yeah, some people it breaks down, all right, but it doesn't break down as much as it breaks down for multiple sclerosis. You understand? Or is it only when someone has a disorder? So that the breakdown is what leads to that disorder. If that breakdown continues happening in different oligodendrocytes in the brain, it will lead to multiple sclerosis. All right? Okay. Don't worry, I'm not talking about pathology. Don't worry, I'll bring out and I'll discuss some of these pathologies for you guys. If you're interested in it, you can always drop me a comment in the comment section and we'll discuss it. All right. Now, for multiple sclerosis, this is just being superficial. For multiple sclerosis, the onset is between 20 and 40, right? And it kills them around 25 to 30 years after diagnosis. You understand? Now, it's usually autoimmune disease triggered by different some viruses. Now we have Tay-Sachs. It's a degenerative disorder of the myelin sheath. Also, number 2 is Tay-Sachs. Yeah, Tay-Sachs disease. All right, it's just a hereditary disease of infants. All right, usually infants that are from the Jewish ancestry. All right. No, it's majorly the CNS. Yes, it's majorly the CNS. That's why we are very specific about oligodendrocytes. Yeah. Okay, so for this Tay-Sachs is when glycolipid accumulates in the myelin sheet. This time around, the myelin sheet, you see, this guy is not a problem, but what is a problem is that things accumulate on that myelin sheet. So when it accumulates on the myelin sheet start acting abnormally, all right? It has an enzyme that called lysosomal enzyme, which will start breaking down that myelin sheet. So when you have abnormal accumulation of glycolipid, right, we call it GM2, abnormal accumulation on the myelin sheet, it leads to what? Tay-Sachs disease. That's just the simple way you know about it. So gangliosides accumulate. And what does it lead to? It leads to blindness. A lot of people that are blind today is just because of this accumulation of that disease, glycolipid. And this glycolipid has high tendency of accumulating when this patient is from Jewish ancestry. That's the first question they will ask you in the hospital. That's the first question. All right. So blindness, loss of coordination, dementia, these are the things that can emanate. All right. Please, when it's time for me to stop, please, you guys should tell me. All right. Because you know me, I can just continue talking for the next 5 hours. Oh, God. Okay. So we have unmyelinated nerve fibers. All right. Unmyelinated nerve fibers. We've already discussed about the myelinated ones. These are unmyelinated nerve fibers. You look at it up later. Now, let's talk about speed of conduction. Speed at which a nerve signal travels along surface of a nerve fiber depends on two factors. Pay attention. This thing is very important. This speed depends on two factors. One is the diameter of the fiber. Another one is what? The presence of or absence of myelin. If there is myelin, it will go faster. Chicken up. If it has large surface area, it will go faster. Chicken up. So large surface area, myelin sheet presence. Large surface area, myelin sheet presence leads to very fast conduction. Okay. Noted, right? So these are conduction speed, right? The small unmyelinated conducts at 0.5 to 2. You understand? M per S. Small myelinated 3 to 5. You understand? Small, because it's myelinated, it's now faster. This one is large, myelinated is the fastest. You understand? 120 minutes per second. Very, very fast. All right. So slow signals sent to the GI tract where speed is less of an issue. So most slow signals goes to the GI, but the fast ones go to the skeletal muscle. You see how I'm talking? I can speak very, very fast. No, you don't need that. No, you don't need. What you really need is to know is that fast ones goes to the skeletal muscle, which is common sense, which makes sense. Think about it. You see how I'm moving my hand? I can keep moving my hand. This is skeletal. You see how I'm talking? I can do it. That's skeletal. But your stomach, your GI rather, cannot do that. It takes it time. Peristalsis. Have you seen peristalsis video? Slow peristalsis. All right. So you don't need to know the time. It's just for you to appreciate the entire thing. All right. So let me fly now. Let me fly. I know I'm behind schedule. This electrophysiology. I think I'm even faster virtually. I think I am. I think I am. So you can read it or read up this at your leisure. This is history. I hardly do histories. Now, you need to know about nerve growth factor, NGF. All right. Sometimes I'll mention NGF. They are proteins secreted by a gland, or muscle, or glial cells and picked up by the axon terminals of the neurons. Right. What they usually do, nerve growth factor, what they usually do is to prevent apoptosis. What's apoptosis? Cell death, programmed cell death in growing neurons. They don't want those growing neurons to die. All right. And also they enable growing neurons to what? Make contact with their targets. Extremely important. There is nerve growth factor. You need to know nerve growth factor. All right. Okay. Then this is talking about electrophysiology, how current moves from here to here. Yeah. So this basis, electrophysiology is the study of cellular mechanisms for producing electrical potentials and currents. It's the basis for neural communication and muscle contraction. All right. Makes sense. It's very, very important. Listen, let me tell you, let me just throw a little light on this thing. All right. The RMP for neurons is minus 70 MV. That's resting membrane potential. All right. What happens? That's minus 70 MV. That's negative neurons. Negative. That's RMP is negative. All right. Now, usually the major cation in the cell. Remember, in intracellular cation, the major one is what? Potassium. The major extracellular cation is what? Sodium. So, despite the fact that it is minus 70, potassium is still inside it. Mind you, potassium is still inside it. A lot of potassium, despite the fact that it's minus 70. Now, for action potential to occur, what happens? The gate has to open, the sodium gate has to open. First of all, sodium. Sodium will rush inside, inside the cell. A lot of sodium will rush inside the cell, raising it up to what? To positive. A lot of potassium will go outside, outside, taking it to what? To negative. A lot of sodium will do what again? Enter inside, taking it to positive. Potassium will go outside again, taking it to negative. So that up, down, generates what? Action potential. Chicken. So it's usually the interaction between the sodium and the potassium. And this thing happens in the membrane. We are 48. Tell them to come back. Oh. Okay. Now let's discuss it properly. The resting membrane potential exists between unequal electrolytes distribution. We already know this, all right? Now the ions diffuse through the concentration. What are the ions? Sodium and potassium, all right? They pass through the cell membrane. Remember, the nerve cell is a cell. So it has a cell membrane, all right? A plasma membrane. So they diffuse through it, all right? Selectively permeable membrane, all right? Leading to electrical activity. So this is potassium. What did I tell you? Potassium has the greatest influence on RMP, resting membrane potential. Plasma, why? Because it is intracellular cation. So plasma membrane is more permeable to potassium than any other ion. What happens to, about potassium? Potassium can leak out until electrical charge of cytoplasmic anions attract it back in, and equilibrium is reached. All right. Potassium is about 40 times as concentrated in the ICF. You see what I was telling you? They will ask you, what is the major ICF electrolyte? That is potassium. Potassium. ECF is sodium. Sodium comes from outside. Potassium goes, out. Never push potassium. It's just like, do you remember in urinary system when we talk about PTH? PTH. We said PTH is potassium tracing hormone. That's what we said, potassium trashing hormone. So it pushes phosphate trashing hormone, rather. It pushes phosphates out, not potassium. People make that mistake. They always call it potassium. I also do that mistake a lot. But that's by the way. This thing you talked about pushing out potassium reminds me of that. Okay. So we have cytoplasmic anions, you understand, that cannot escape due to the size, such as what? Phosphates, sulfates, small organic acids, proteins, ATP, and RNA. All right. Now this is sodium. You see what I was telling you? Sodium is about 12 times as concentrated in the ECF as the ICF. So sodium is the major extracellular cation. Potassium is the major intracellular what? Cation. All right. It's as simple as that. I think that's the information you need to know from this particular place. So sodium potassium pump moves 3 sodium and 2 potassium. 3 sodium, 2 potassium. All right. Okay. So going up, going down, going up, going down until it forms what? Action potential. So look at it here. This is the diagram for you guys to understand. What's the time? 4:02. Okay. This is the diagram. All right. For extracellular fluid, ECF, sodium is what? 145. If we are checking electrolytes, guys, if we are checking electrolytes, so you come to the clinic, for example, and we do you serum electrolyte, urea creatinine, which electrolyte are we checking? Are we checking the ICF or the ECF? Everybody, tell me. Which electrolyte? Oh, we say, oh, this person is depleted of electrolytes. Is it ICF or the ECF? Which one are we checking? Everyone, is it ICF or the ECF? When you come to the clinic, they say, oh, this person is losing blood. Maybe someone that is losing a lot of blood. You understand? And we want to check the electrolytes of that individual. Are we checking the ICF or the ECF? Let's go. Let's go. We have 50 people here, so I need at least 30 people to give me comments. You guys are absolutely correct. Okay, I remember most of you work in the clinic. Yes, so we are checking the ECF, not the ICF. So mind you, that's why you see normal range for sodium in the ECF is what? 145. Why? When you're talking about the ECF, that is extracellular fluid, you're talking about plasma. You're talking about interstitial fluid. Do you understand? When you're talking about ICF, you're talking about the cytosol. All right, so I need you guys to get this information. It's extremely important. So if you look here now, you see sodium, because sodium is more in the outside, sodium is jumping in. Potassium is more in the inside, potassium is what? Jumping out. So this interchange, you understand? 3 sodium comes in, 2 potassium goes out. 3 sodium comes in, 2 potassium goes out. So at the end of the day, the internal environment becomes what? Positive. Action potential will occur. That's what they are trying to tell you. Because there is more positive ions coming in than going out. Potassium is still positive. Sodium is still positive. So when potassium is going out, this place continue being negative. But sodium will keep coming in. At the end of the day, when you balance the equation, you see that there are more sodium that entered than potassium that left, because it is 3-1. That's how action potential is generated. All right. So there's something called local potentials. All right. Local potentials, that's when there is this change in membrane between nearby cells. All right. Nearby cells that is stimulated. Just nearby cells. Very important. So there are different neurons that can be stimulated by this. We have the chemical, light, heat, or mechanical disturbance. So it's still, at the end of the day, all of them is still all about sodium and potassium. Sodium and potassium. Opens sodium gates and allows sodium to enter the cell. Entry of positive ions make the cell what? Less negative. And that less negativity is what? Depolarization. Depolarization. So the thing will change from negativity to what? To 0. So sodium entry results in current that travels around cells, triggering zones, right? So it's very, very important for you guys to master this. Sodium enters, changes from more negative to what? Less negative. All right? So these are the properties of local potential. We have local potential and we have action potential. Mind you, we are still in local potential. We are still in local potential. So these are properties of local potential. They can be graded, they can be decremental, they can be reversible, they can be excitatory or inhibitory. They will ask you, one of the following is not a property of local potential. They can be graded, meaning that, oh, we can see a smaller local potential or a larger local potential. They can vary in magnitude. They are not the same strength. That's what we mean by graded. They can be decremental, means that they get weaker as they go further. So for example, if I shake this thing now, the part I shake will vibrate more than the part. And when the wave keep going outwards, right, it will drop. That is local potential. Local potential does not go up. Instead, it comes down more. That's what you need to know. It's decremental. So the voltage diminishes. Then it is reversible. Sometimes if stimulation ceases, the cell quickly returns to resting membrane potential. All right. Remember local potential. The cell resting membrane potential is minus 70 MeV. All right. Now sodium rushes in. It changes from minus 70 to 0. That's what we call local potential. All right. Graded, decremental. It can be reversible. It can go back to resting membrane potential quickly. All right. And sometimes it's either excitatory or inhibitory. Okay. I hope this is the diagram for you to understand. The local membrane potential moves down this way. Okay. Now let's talk about action potential. Action potential is very, very important. It occurs only when there is high enough density of voltage regulated gates. Extremely important for you guys to know this. Very important for you guys to know this. All right. Neurosoma cannot generate an action potential. It's not possible. It cannot generate an action potential. Trigger zones, you understand, is where action potential are generated. Trigger zones is where action potential are generated, not in neurosoma. So if they ask you this question, you should be able to understand it. You should be able to at least remember this particular statement. All right. Okay. Okay, so depolarization must occur. It must get to the threshold, right? We have the critical voltage about minus 55 MV required to open voltage regulated cells. All right. All these things happen before you know it, it gets to 0. So as membrane rises above 0, remember we say in 0, we are still talking about local potential, right? It suddenly peaks to plus 35 MV. Slow potassium channels open and there's outflow of what? Potassium, leading to what? Repolarization. Repolarization. All right. Look at the diagram here. Look at it, guys. It's what is the resting membrane potential. When it is around 0, that's local potential. It can still go back. But when it gets to 35, it forms what? Action potential plus 35. Look at it here. After action potential, it can now go back down. All right. Go back down. Sometimes it go beyond minus 70. Yes. It's the exon hillock. So sometimes it goes less than minus what? 70. Maybe something like minus 90. That minus 90, we call it what? The region of what? Hyperpolarization. All right. So when it's going up from minus 70 upwards to positivity, we call it what? Depolarization. When it is coming down back to negativity, we call it what? Repolarization. And everything going up and going down here leads to what? Action potential. All right. So this is how sodium and potassium interact. You understand? This is their interaction, going up, coming down. All right. Okay. This is the laws you need to know for action potential. All right. Very, very important. It can spike and it happens very, very fast. It spikes, it happens very, very fast. Characteristics of action potential, it follows all or none law. All or none law is either it gets to this point or it doesn't. Like if it gets to this point, action potential must be formed. If it doesn't get to this point, it can never form action potential. That's the meaning of all or none law. All right? Follows all or none law. If threshold is reached, the neurons fires at maximum voltage. If threshold is not reached, neuron does not fire. Simple. It is not decremental. You see the difference between action potential and local potential. It is not decremental. It doesn't get weak, no matter the distance. So an action potential that's produced here, we can travel a long distance and still produce that effect in the nerve and transfer to the muscle fiber and lead to that contraction. That's action potential for you. Non-decremental. Follows all or none law. It is what? Irreversible. Once action potential has been stimulated, it does not go back. But local potential can go back. Local potential is decremental. You need to know the difference between these two. Extremely important. All right? This is action potential in real time. So you look at it later. Now, there's something called the refractory period. The refractory period. This is the period where it is difficult or impossible to stimulate a region of a neuron to fire again. Like in reproductive system, you talk about refractory period as well. Here, you talk about a refractory period as well. There's nothing. Like in reproductive system, when I think no, it wasn't your class. I taught reproductive system. I was telling them that when there's a level a man gets to, and the man's stuff does not rise anymore until a certain period of time. So that thing is called, in reproduction, it's called refractory period. Here, it's also called refractory period. Remember that. We even have two types of refractory period. We have a relative, we have the absolute. Relative is, oh, you try, and finally it started again. But absolute is, it's not happening. It does not what? Happen. All right? Oligodendrocyte. It doesn't what? Happen. Okay. So yeah, this is the absolute refractory period, relative refractory period. Okay, look it up later. So you see, this absolute refractory period, at this period, there's nothing, no stimulation can cause action potential anymore. All right? Remember this diagram. You will see exactly this diagram in reproduction with different terminologies. Okay. I don't know. Should I ask you one or two questions, or should I continue? What do you suggest? Somebody said I should not ask question, I should continue. That was your questions. Somebody say continue. You are enjoying the flow, eh? Okay, fine. Let me give you two questions. This is one. So this is one question. I can give you options if you pay attention. This is one question. The question is, conduction of a nerve impulse would be fastest in which of the following? Would be fastest in which of the following? I need to tell me, is it A, B, C, D, or E? A large diameter myelinated axon. B, a large diameter myelinated, A large diameter myelinated axon, that's A. B, a small diameter myelinated axon. C, a large unmyelinated axon. D, a small unmyelinated axon. And E, a small axon with multiple Schwann cells. Oh boy, I'm supposed to go to the lab. Oh, perfect. You guys are correct. It's a large diameter myelinated. A large diameter myelinated. Extremely important. Okay, look at this question, guys. Very fast, very fast. I know the time is already against me. Which of the following ions has the greatest influence on the RMP, resting membrane potential? Which of the following ions has the greatest influence on the resting membrane potential? Okay, I said which of the following you are expecting options. Oh, we have sodium chloride, calcium, phosphate, and potassium. Sodium chloride, calcium, phosphate, and potassium. Okay. You guys are very hot too. Extremely hot. Okay. Final question before I continue. And this question is a little tough, eh? So this is the question here. In a neuron, the opening of the sodium gates typically leads to this. The question. This is I'll post the options. This is A. This is let me post the options. This is A. This is B. Incoming missile. This is B. Then this is C. This is D. This is E. Okay, let's go. In a neuron, opening of the sodium gates typically leads to A, repolarization of the plasma membrane, B, hyperpolarization of the plasma membrane, C, depolarization of the plasma membrane, E, drifting of a plasma membrane voltage toward a more negative value, and E, plasma membrane voltage returning to the resting membrane potential. Answer. Perfect. You guys are correct. It is what? Depolarization of the plasma membrane. Okay. Sorry. Okay, so that's the polarization of the plasma membrane. Yeah, you guys are correct. You guys are extremely correct. All right. So this is talking about conduction, different conductions from here to here, from here to here, from here to here, until it gets to the extreme. If you look at the red, the action potential in progress. Then the yellow is where we have what? The refractory membrane. Then the blue The green is what the excitable membrane. That's how action potential moves. Yes, you guys are extremely correct. Now there's something we call saltatory conduction. Saltatory conduction is jumping. So I need you guys to take time and look at this. All right, nodes of Ranvier contain voltage ions, while myelin sheets, myelin covered internodes contain few. All right. So when sodium enters the cell at a node, its electrical field repels positive ions inside the cell. I need you guys to look at this. I don't want to read it, it will waste my time. But look at this. But I need you to always know saltatory conduction. Saltatory conduction is what? Jumping. Pium, 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 pium. It keeps jumping, all right? So the faster they jump, the faster it is, all right? It jumps pium, pium, from here to here. See it? It enters here, it jumps to this place. From here, it jumps to this place. So it avoids virtually all this place, all right? Virtually all this place, just jumps, pium, pium. So it keeps jumping from one node of Ranvier to another node of Ranvier to another node of Ranvier. Before you know it, it has gotten to its destination. Look at it. Pium, pium, pium. Okay. So that's, by the way, that's beside the point. Now, let's talk about synapses. This synapses is extremely important. Remember, I told you there is a nerve signal can reach its end. All right. We have two things that can synapse. We have synapses between two neurons. Remember, I told you about synapses between two neurons. All right. And we have synapses between a neuron and another a neuron and a muscle. Extremely important for you guys to know this. You understand? So synapses between two neurons. First neuron in the signal part is the presynaptic neuron. So we have presynaptic neuron, which releases what? The neurotransmitter. Now we are talking about neuron to neuron. Neuronal, not neuromuscular. Yet we have neurotransmitter. All right? Okay, remember nerve cells produces neurotransmitter. It's one of their functions. Now the second neuron is the post. The first neuron, where the signal is coming from, is the presynaptic neuron. The next one, where it's transferring to, is what? The postsynaptic neuron. So this presynaptic neuron will send out this neurotransmitter, right? Once the action potential comes down, it will trigger neurotransmitter from the presynaptic. The presynaptic will send it across to the postsynaptic, and this postsynaptic will respond to what? That neurotransmitter that came around. All right. So presynaptic neuron may synapse with the dendrites, neurosoma, or anything. You understand? That's why we have what? Axodendritic, axosomatic, or axoazonic. It can synapse with the dendrites, the cell body, or another axon. So axodendritic, axosomatic, or what? Axoazonic. Very important for you guys to know these things. All right. Now, in the spinal motor neuron, covered by about 10,000 axons, terminals from other neurons, 8,000 ending what? In dendrites, 2,000 ending in what? Neurosoma. So the dendrites win it. If you do auction, 8,000 people are getting dendrites, 2,000 people are getting neurosoma. The dendrites what? Win it. It's as simple as that. All right. That's for the spinal cord. In the cerebellum of the brain, one neuron can have as many as 1,000 what? Synapses. All right. So if you look at this diagram, I'm trying to rush this thing because, like, you need to understand it. If you look at this diagram, this is a soma, this is an axon. All right. It is a presynaptic. Look at the direction of the action potential. It's coming from this particular guy, meaning that this guy is the presynaptic. It's entering this guy. This guy must be what? The postsynaptic. And this is the axon. This is the terminal part of the axon. It's transferring it to what? To the soma of this other neuron. So this is called what? This is called what? The exosomatic. But if you look at this guy here now, what do you see? This is axon transferring to another axon or to another dendrite. Look at it. Okay, this is a dendrite transferring to another dendrite. So this become what? If you look at it very well, this is an axon. Let me see. Sorry, I need to zoom this thing so that I'll be able to everybody focus on it. Okay, look at it properly. What do you see? You see the cell body here. You see the axon here, comes down here. Then this is the cell body. This junction now, this junction between the cell body, the axon, and this cell body is called what? Exosomatic. Look at it here. This is the axon. This is the cell body. This is exosomatic. And this is axon here. This is axon here. This is the axon here, and this is the cell body. This is exosomatic, and this is axon here, and this is dendrite. This is exodendritic. Now, this particular axon goes down here. All right. I hope this thing is not lagging, guys. It goes down here and it merges with another what? Axon here. This is exoaxonic. So I need you guys to know exoaxonic between an axon and an axon, exoaxonic. Now I need you to know axon between an axon and a dendrite, that's exodendritic, and between an axon and a soma, that's exosomatic. Those are the three major things you have. You can see any of these stuffs in your, they can give you this picture in your exam and tell you this is A, this is B, and this is C. So if you are just seeing an axon, this is an axon, and another axon, just know it's an axon axonic. If you see an axon and what? And a cell body, this is known as, this is what? An exosomatic. And if you see an axon and a dendrite, know that this is what? An exodendritic. All right? Okay, let me unzoom it. Let me unzoom it so that, sorry, I've unzoomed. I had to zoom it so that, I hope it didn't lag, so that I'll be able to see it well and you guys will be able to see it well. How many minutes do I have? 2 minutes. Hey, you guys will borrow me like 10 minutes, so please. I really want to complete this thing so that next this thing we can start up. Say 1 minute, please. Can you add 0 to the 1? Let it be 10, please. So 30 seconds, please. Okay. So now if you have something, you can actually jump off because I'll take another extra 15 minutes to finish up this slide. Then if you finish, you can just go and look at it so that next time we will go straight to the brain and the spinal cord. All right. Remember, this is just an introduction of the entire neuron. So I really need to finish this. All right. Okay. Let me see. Okay, so look at this part. This is structure of a chemical synapse. You look at this part. This is, you see, this is the end part of the axon. This is the neurotransmitters being released by exocytosis. Fine, just for you. Thank you. Thank you. This is the ending part of the, this is the neurotransmitters inside the vesicles, right? You see how the neurotransmitters comes down. Look at it. Comes down. You understand? This area is the synaptic cleft. This space is the synaptic cleft. This is the vesicle. They may ask you, how does these vesicles bring out their contents? They bring out their contents through what? Through what? Exocytosis. Extremely important that you know this. Right? Now, when they release these neurotransmitters, this, you see, the neurotransmitter comes to this particular surface. Oh, I should make it bigger. Okay. Yeah. So this is what we are talking about. This is one neuron. This is another neuron. All right. This is how it works. This is one neuron. This is another neuron. So this is presynaptic, and this is what postsynaptic. See, this is where we made it bigger. This part is where it was zoomed. So this space is known as what? It's synaptic cleft. Any junction, there must be a synaptic cleft. Now, remember, the action potential comes down, triggers calcium. Calcium will bust these guys up, these vesicles up. These vesicles will pour out through what? Exocytosis. I'm repeating this exocytosis because of how important it is. Now, when these vesicles burst, it brings out exocytosis. That the content is what? Neurotransmitters. Now, the neurotransmitters will do what? We come. This red thing is the receptors, neurotransmitter receptors. The neurotransmitters are chemical messenger. We call them first messenger. They are always the first messengers. Now, they lap on this red thing. This red thing is the what? The receptor. When they lap on this red thing, they will now cause conformational change inside this other neuron, activating the action potential of this neuron, and it will keep moving until it gets to the muscle. You need to understand this, all right? We will still discuss it, but you need to understand this. It's extremely important because you see these receptors. If this receptor has a problem, this is what leads to a lot of problems like myasthenia gravis, like, when we have inadequate receptors. You understand? When these receptors begin dying, maybe as a result of autoantibodies, when they start dying, it leads to myasthenia gravis in a lot of patients, honestly. And this same thing is also what happens for diabetes type 2. Remember the difference between type 1. Type 1 is not producing enough. Type 2 is that they are producing enough insulin, but the insulin could not match to the receptors. It is the same receptors that we are talking about. These are receptors. So anytime you produce a chemical messenger in the body, it must match to a receptor for there to be a conformational change. Sorry, I'm being fast. All right? For there to be a conformational change. So I hope you get that. Okay. Have a nice one. Later, make sure you come and watch this. Have a nice one. Okay. So Neurotransmitters like acetylcholine, we have amino acid transmitters, acetylcholine, amino acid transmitters like glycine, glutamate, aspartate, and aminobutyric acid. That's GABA. All right. So these are neurotransmitters I need you guys to know to prepare for your exam. We also have monoamines. We also have purines. All right. So later you take your time and look at this diagram. Let me see if there's any other thing you need to know. So yeah, we have the excitatory cholinergic responses, the inhibitory, and we have the adrenergic. So we have, thank you so much. We have the cholinergic, we have the excitatory, you have the inhibitory rather. All right. Then we have the cholinergic, we have the GABAergic, we have the adrenergic. The adrenergic is always excitatory. The cholinergic is always excitatory. They may ask you in your exam. Got to run. It's fine. All right. Have a nice one. So the cholinergic and the adrenergic are both what? Excitatory. They may ask you which one is excitatory in your exam. Cholinergic, adrenergic is excitatory. But GABAergic is what? Inhibitory. I think that's what you need to know for this level. So this is everything about cholinergic. You can read it up later. Trying to move past this. I'll still review this at the end of the day. Okay. So this is cessation of signals. All right. This is postsynaptic potential. Okay, I think we are pretty done. I'm trying to check if there is something else we need to learn. I think we are pretty done. Okay, so yeah, this talks about memory, and immediate memory and long-term memory. All right. I think we are pretty done. Okay, then this talks about pathology, Alzheimer's disease. Okay, I think we are pretty done. So if you learn it to this neuromuscular junction transmission, then you are good to go. The only thing I need you guys to study later on is to learn these different types of synapses. All right, this excitatory, the inhibitory, and another excitatory. You should know that cholinergic and adrenergic are always what? Excitatory. So if somebody is there's a excitatory, that's like, for example, if there's contraction going on, right, it's mostly cholinergic or adrenergic. If there's inhibition, it is usually what? GABAergic. You should know that. So what you need to know is the chemicals that makes it happen. All right, how calcium works, how potassium works, and how sodium works. It's always the three of them that causes the entire changes. So I need you guys to learn this at your leisure. So go home and study this. You can screenshot. For people that don't have my slide, you can, this particular slide, you can screenshot this. So you read it up to know the what calcium does. All right. I told you that calcium causes exocytosis, breaks down the vesicles to release what? ACH. That's the neurotransmitter. That's what calcium does. All right. And once ACH is released into the postsynaptic cleft, it leads to what? Sodium and potassium going up, going down, going up, going down until action potential is produced in the postsynaptic what? Membrane. All right. So cholinergic, excitatory. Then this is inhibitory. GABAergic. GABAergic works with usually chlorine. Always chlorine. Chlorine will enter the cells, and chlorine is negative, right? It enters the cells. Once chlorine enters the cells, everything becomes what? Negative. Everything becomes negative. That's why GABAergic is what? Inhibitory. So if you know that cholinergic is excitatory, you know that cholinergic works with calcium, potassium, and sodium. Check it out. Everything positive. It's excitatory. It wants to excite. But GABAergic is inhibitory. It works with chlorine. Remember that. You understand? Remember that. All right? And GABA receptors, right? Which are chlorine channels. It's as simple as that. And chlorine is negative. Now, when you talk about adrenergic works with the norepinephrine, or we call it what? Noradrenaline. You need to read this, all right, so that you understand how it works. Remember, these things are first messengers. They are neurotransmitters. Neurotransmitters are always first messengers. What they usually do is to trigger the receptors, and the receptors will cause a conformational change inside, all right, leading to either excitation or what? Inhibition. You need to know that, all right? So this is how it works. For adrenergic, this is how adrenergic works. Once they look at this red thing is the receptor. You see how the receptor works? So immediately the neurotransmitter matches on the receptor. What happens? The receptor becomes what? Activated. How does it get activated? There is something we call the G protein. The G protein causes the adenylate cyclase to be activated. Then the adenylate cyclase will activate the ATP. The ATP will activate the CAMP. This CAMP is what we call the second messenger. The first messenger is what? The neurotransmitter. The second messenger is the CAMP. And how does it happen? Specifically for adrenergic response, what it usually does is it will activate the G protein. The G protein will activate the adenylate cyclase. Adenylate cyclase will trigger ATP and convert it to CAMP. CAMP is the second messenger, and the CAMP will now do every other thing. And what are the effects of CAMP? Everything. Everything. Look at it. Metabolic changes, enzyme activation, enzyme synthesis, gene transcription, everything is done by the CAMP. That's why we say that the CAMP is the second messenger, but the first messenger is the neurotransmitter. You need to remember that. All right? Okay. So that's that. Then at the end of that, it leads to cessation of signals, and we are good to go. All right? So this is what happens in the postsynaptic potentials. You talk about the end-plate potentials and others. Just read up this. All right? But we've already discussed the major thing. Let me see if there is something else. Okay, so as I mentioned initially, go and read up memory and synaptic plasticity. All right. So thank you guys very much. Assuming we are still many here, assuming we are up to 10 here, I can throw you 1 or 2 questions for the benefit of people. Are we up to 10 here? If you are still here, say yes. I want to throw like 2 to 3 questions before I release you guys. If you are here, say yes. Let me see how many yeses I get. So the people that are not here can actually come back here, all right? So because since I made out this, I'm going to the lab, but since I have up to 10 to 15 minutes, I can easily use it to, discuss with people that are available so that others can look at it later. Okay. We have up to 30 people. Are you serious? It says 31 people are. Okay. Then let me give you this question. I'll give you like 3 to 5 questions, and we can now end it. Okay, look at this question. Look at this question, guys. But I'll read it out for people, the benefit of people that are not here. Which of the following will cause the plasma membrane to hyperpolarize when at its RMP? Let me give you the options. This is A. Okay, take care. This is A, an inflow of sodium. This is B, an inflow of potassium. This is C, an inflow of calcium. This is D, an inflow of chlorine, and this is E, an outflow of chloride. So what we call that hyperpolarization? Yeah, it's the 30 people. All of you that said yes, I need you to give you the answer to that. Which of the following will cause if you don't know it, just write not sure. Which of the following will cause the plasma membrane to hyperpolarize when at its RMP? Keep going, keep going, keep going. You guys are very correct. You are extremely correct. The answer, you guys are extremely correct, all right? The answer is D, which is an inflow of chloride. Because when there is an inflow of chloride, it will look at the hyperpolarization. If you look at, yes, you are correct. If you look at, you see the hyperpolarization? This is the hyperpolarization. It's negative, but it's now more negative. Why? Because someone said we should do virtual. Yeah, I found out that it's very fast, but like I like to see your faces. Sorry. Okay. Now we'll be doing the revision many times. Look at this very fast, guys. That answer is hyperpolarization is D. Look at this one very fast. Local potentials are dash, meaning they vary in magnitude according to the strength of their stimulus. Are they stable? Are they graded? Are they self-propagating? Are they excitatory? Are they inhibitory? All right, see you later. Take care of yourself. So what's the answer? Local potentials are dash, meaning they vary in magnitude according to the strength of the stimulus. Stable, graded, self-propagating, excitatory, and inhibitory. Yeah, it's automatically saved. Anytime you come to the live, you see it. That's why I'm reading out the questions so that when people come, because they may not see the comments. So when they come, they will listen to the questions. Yes, you guys are very, very correct. Local potentials are graded, meaning that they vary in magnitude and they vary in magnitude according to the strength of the stimulus. Yes. Let me give you two more questions and I'll leave you guys, all right? Yeah, they are graded, of course. Which of the following is most likely to cause a rapid depolarization? Which of the following is most likely to cause a rapid depolarization? Yes, that answer is graded. You're correct. Which of the following will most likely cause rapid depolarization? This is A, that's opening of chloride channels. B, opening of sodium. And C, opening of potassium. Let me put it in the box. C, opening of potassium. D, inactivation of sodium channels. D, inactivation of sodium channels. And E, closing of sodium channels. Let's see. Let's see. Which of the following is most likely to cause a rapid depolarization? Answer. You know why I'm focusing very much on this is because most of your drugs works this way. Okay, you guys are extremely correct. Sodium channel is open. Okay, look at this question, guys. Look at this question. Most drugs open sodium channels. Not most, but there are a lot of them. There are the ones that open calcium. In short, each of them has drugs that works for them. Depends on what we are trying to target. All right. Look at this question. Dash is an action not dash in an action potential. Which event directly follows repolarization? Which event directly follows repolarization? Let me read the option. A, pay attention so that I get the option. A, threshold is reached. B, hyperpolarization of the membrane. C, depolarization of the membrane. D, hypopolarization of the membrane. And E, the resting membrane potential is reinstated. A, threshold is reached. B, hyperpolarization of the membrane. C, depolarization of the membrane. E, hypopolarization of the membrane. And D and E, the resting membrane potential is reinstated. Let me see. Hyperpolarization. A, threshold is reached. B, hyperpolarization. C, depolarization. E, hypopolarization. And E, resting membrane potential is reached. Let me see. Keep going, guys. There's one hyper. Yes, you're correct. You guys are very correct. It's hyperpolarization of the membrane. Okay. Last question for today. Let me give you last question. Final question. Please, I need everybody. You are more than 20. I need everybody to answer this question. During the absolute refractory period, which of the following is true? I'll give you the options, all right? Once I give option, tell me A, B, C, D. This is A. This is B. This is C. This is D. And this is E. So during the final question, during the absolute refractory period, which of the following is true? A, if a neuron reaches threshold, the action potential goes to completion. B, the neuron fires at its maximum voltage if a stimulus depolarizes the neuron to threshold. C, the signal grows weaker with distance. D, it is possible to trigger a new action potential, but only with an unusually strong stimulus. And the final question option, that's E, no stimulus of any strength will trigger a new action potential. What's the answer? What's the answer? Okay, you guys are very correct. The answer is E. No stimulus of any strength will trigger a new action potential. That's what absolute refractory period. So that's for our first neuro one, right? You guys are the best. And I really appreciate the fact that all of you, many of you, not all of you, by the way, but that's fine. That's fine. The others that are not here due to their schedule can always look it up in the streaming and get more information about it. And please, if you can, try to see the other videos. It's extremely important. I will post the link. I probably should post the link on your Connect, so that you'll be able to get more information. Thank you guys very much. You guys are the best. I'm so far happy that we are able to finish this today so that we can focus on other things next time. Thank you so much. Oh, I appreciate. One bottle of water for you. Yeah, good one. Have a good day. Yeah, take care, guys. Yeah, thank you so much. Have a nice one. Take care, guys. You guys are the best. All right. Take care. Thank you. Thank you. Thank you. Yeah, you too. Okay, so I'll add this to this assignment. Fifty concepts. Mm. Yeah, thank you so much. Have a good one. Take care. No, I know. Don't worry. I know. I know. Don't worry. There's no attendance today. Take care, guys. Bye bye. Let me end the stream now.